Nucleic acids are some of the largest molecules that we do find inside of our cells. And these are going to be large polymers made of monomers that are called nucleotides. As far as the scale goes, in some cases we will have hundreds of millions of these nucleotides linked together. And that's why we say these are the largest molecules that we do tend to find inside the cell. There's two different categories for our nucleic acids, and these are probably molecules that you've heard of before. This is going to include DNA, and then it's also going to include RNA. If we look at the nucleotide structure a little bit more, here we have it. The nucleotide is made of three components that are hooked together. We have a phosphate group, we have a sugar component. We like to call that a pentose sugar because it's got 5 carbons in it. And then we also have a base component that is going to be included here. Those nucleotides, they are going to be connected together to make those really large nucleic acids. But if we look at the nucleotide structure a little bit more and talk about the variety of the pieces, the phosphate group is always going to be the same. There's no variety to that. But the sugar component, that we have two different options. The sugar component can be what we call deoxyribose, and that is what we find in DNA. And that's really what the D in DNA stands for. And then ribose is the other option, and that is what we find in RNA. And that is what the R stands for. So DNA and RNA, their nucleotides will have a different sugar. Those sugars, it's really a minor difference between them. The only thing that's different is this little area here that's shaded in pink or that I've circled. And then the other thing that varies with our nucleotides is the base component. The base component has 4 different options that can be found there. You can see those over on the side of the slide. We have some that are a little larger, and we call them purines. So the purines have 2 rings that are fused together. And then we have pyrimidine components, which are smaller, and they are just a single hexagon ring. What we find in RNA and what we find in DNA is a little bit different with the base component as well. Now, if we take these and we start linking them together, they're going to be joined together with synthesis reactions, and when they're joined together, we get the super long polymer. The sugars and the phosphates will be what actually links together. So we'll see if you just look down here, it's sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on the whole way down. And so we call that a lot of times the backbone of the chain or of the molecule. And then notice that the bases over here are just kind of sticking out to the side. The order of those bases is very specific. And that is what makes my DNA sequence different from your DNA sequence. So that is our sequence of the DNA. So every individual has a different sequence there, and that sequence is also interpreted by our cells to make specific proteins and run specific activities in our cells. So here we have just another look at DNA. DNA, the structure itself is that it is made of 2 strands. So 2 strands like we just described that are going to be hooked together. And they are hooked together by hydrogen bonds. The hydrogen bonds are located down the middle of the strands, and they are found between the bases in the 2 opposing strands. Another important thing about the structure is that the 2 strands run in opposite directions. So they kind of run like this. And we can see that here because the ends are labeled differently from each other. Another last thing, which I think everyone's pretty familiar with already, is that it's a double helix, and that means that not only is it 2 strands, but the 2 strands are going to be twisted to form a helical structure. DNA is ultimately the genetic material for all organisms. That DNA will be organized with some proteins a lot of times, and we'll call the larger structures chromosomes. So DNA is what makes up our chromosomes, but this is the information that our cells use to run all of their activities. This is the information that our cells pass on to their offspring when we talk about genetic material or hereditary information. The last molecule to discuss is what we call adenosine triphosphate, or ATP. The reason why this molecule is so important for us to introduce here is because it is the main short-term recyclable energy supply molecule for cells. So when we talk about cells needing energy for a reaction, they're usually going to get that energy from ATP. So if we look at ATP and think about it as a battery, this is what we have. It's a rechargeable battery. When it is not charged up, we call it ADP. Okay, so notice that ADP is just a partially charged battery. But if we do charge it up, so if we pack some energy in there, we're going to call it ATP. So ATP is a fully charged battery, and this can be used to power a variety of reactions. And so the type of reactions that would be using ATP as a power source would include our synthesis reactions. So they had energy going into them as a reactant. They're going to get that energy primarily from a charged up ATP molecule.